Meiosis, life's greatest miracle. Meiosis is a type of cell division in sexually reproducing organisms that reduces the number of chromosomes in gametes. In humans, body cells are diploid, containing two sets of chromosomes, one from each parent. To maintain this state, the egg and sperm that unite during fertilization must be haploid, with a single set of chromosomes. During meiosis, each diploid daughter cell undergoes 2 rounds of division to yield 4 haploid daughter cells, the gametes. This division of cells has 5 key steps: prophase, anaphase, metaphase, telophase, and finally cytokinesis. The first step in this important process is prophase. Prophase is the first phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into 2 identical daughter cells. During prophase, the complex of DNA and proteins contained in the nucleus, known as chromatin, condenses. The chromatin coils and becomes increasingly compact, resulting in the formation of visible chromosomes. Chromosomes are made up of a single piece of DNA that is highly organized. The replicated chromosomes have an X shape and are called sister chromatids. The sister chromatids are pairs of identical copies of DNA joined at a point called the centromere. Then, a structure called the mitotic spindle begins to form. The mitotic spindle is made of long proteins called microtubules that begin forming at opposite ends of the cell. The spindle will be responsible for separating the sister chromatids into 2 cells.
Metaphase, the second stage during the process of cell division in meiosis. Normally, during this stage, individual chromosomes are spread out in the cell's nucleus. During metaphase, the nucleus dissolves and the cell's chromosomes condense and move together, aligning in the center of the dividing cell. The spindles act as fishing rods, reaching out and grabbing the chromosomes. The next phase after this is anaphase.
Now for the best part of meiosis, anaphase. The preparations are complete, the chromosomes are replicated, and the division is ready to occur. This is an important step in meiosis due to the opportunity for genetic variability, with parts of each homologous chromosome crossing over and essentially fusing into one another. Now for a more in-depth explanation.
Anaphase is one of, if not the most important stage in the process of meiosis, since it is when the division of the cell actually occurs. In anaphase, the spindle fibers, attached to the chromosomes, which are lined up in the center of the cell, pull the chromosomes apart at the centromere, perpendicular to the chromatids, until there are 92 separate identical halves. During this process of separation, genetic variation also occurs, with parts of each chromosome crossing over to the other, making each chromosome afterwards a mixture of the 2 parent chromosomes. This is a very useful characteristic in sexual reproduction, making each child different from each of their parents, since it can help in natural selection, making the species stronger and more adaptive to their surroundings. After the initial separation of chromosomes, there comes telophase, which further divides the cell in 2.
The next and the final stages of meiosis 1 are telophase and cytokinesis. In telophase 1, the nuclear envelope begins to reform around each end of the cell where the chromosomes and chromatids are present. Each side of the cell contains only 1 chromosome and 2 chromatids. Each cell is technically haploid because of this, and also, by this point, most of the spindle is dissolved. Telophase 1 in meiosis differs from mitosis in the fact that the parent chromosomes rather than the sister chromatids are separated, and after the completion of telophase comes cytokinesis. In cytokinesis 1, the cell follows a similar process to mitosis, where the cytoplasm pinches and ultimately divides the one larger cell into 2 smaller daughter cells. After cytokinesis 1 is completed, each daughter cell will function independently to complete meiosis 2, the second stage of meiosis.
Meiosis 2 is the second stage of cell division that occurs in sexually reproducing organisms following meiosis 1. Starting with prophase 2, following the initial division of meiosis 1, the nuclear envelope disintegrates, allowing spindle fibers to extend and chromosomes to further condense into distinct sister chromatids. Transitioning into prometaphase 2, spindle fibers attach to the centromeres of the chromatids, guiding their movement towards the center of the cell. Metaphase 2 sees the alignment of the chromosomes along the metaphase plate, positioned by the tensing of spindle fibers. Anaphase 2 marks the separation of sister chromatids as cohesion proteins are cleaved, repelling them towards opposite poles. As telophase 2 ensues, chromatids reach the poles. Chromatin begins to decondense and nuclear envelopes reform. Finally, in cytokinesis 2, the cytoplasm divides, yielding 4 haploid daughter cells, each with unique genetic composition, including the intricate process of meiosis essential for genetic variation in sexual reproduction.
And with that, the process of meiosis is complete. These cells will continue to divide and grow in a different process known as mitosis, creating what we are today, human beings. Thank you for listening, and I hope to shed more light on the essential process of meiosis and the creation of life itself.